Development of a biologic begins by identifying the protein of interest and isolating the gene which codes for it. The gene is then joined to a piece of DNA called a plasmid vector, creating an expression vector used to produce the desired protein. The expression vector is inserted into special cells adapted for producing proteins. Through a process called transcription, the cellular machinery reads the gene, creating a molecule called messenger RNA, or mRNA. Further machinery uses mRNA to produce a protein, which is folded into a unique 3D structure. The protein undergoes further modifications, such as the addition of sugar groups, contributing to the structural and functional diversity of proteins. The protein is then secreted into the media and harvested to prepare the biologic. Cells producing the protein of interest are grown in bioreactors, which are devices used in commercial production of proteins. Initially, cells from frozen stocks are grown up as cultures in small flasks, and then the cultures are successively scaled up in larger vessels until they reach the large bioreactors, which can be up to 10,000 liters. Variables, including temperature, pH, nutrients, and oxygen levels, are continually monitored and optimized within the bioreactor. In suboptimal conditions, for example, when temperature is too high, the cells will produce less protein or start to die. Controlling this environment helps to ensure proper cell growth, metabolism, and various protein modifications required for proper protein folding and function, and therefore helping to ensure high-quality product.
Hi, I'm Ryan. I work here in manufacturing. As you can see, I'm now wearing my plant uniform. One reason we wear these plant uniforms is to assist us in helping keep the facility clean. We're now going to go down into one of our production areas. Follow me. And the reason we do this is to assist us from keeping contaminants from entering into our manufacturing facilities. In an environment like this, the greatest potential for contamination actually comes from humans. This is why we take great care wearing the appropriate apparel within the clean room environment. This area is also equipped with HEPA filtration. These allow us to do full air exchanges in this room approximately every 2 minutes, assisting us in keeping proper clean environments for where we produce our medicines.
I'm Michelle. I've been with Amgen for 13 years. I'm a frontline manager here in the cell culture area. The main purpose of the cell culture process is to scale up to higher and higher volumes, ultimately resulting in higher cell densities, which means when we transfer to the production bioreactor, we're able to produce protein. We start our process with just a 2 ml vial of cells, and we continue to scale up to bigger and bigger volumes. This is a 50-liter cell bag bioreactor. It's one-time use only. We run this process for 3 days, and we continue to scale up.
Hi, I'm Tracy, and welcome to purification. At this point in the process, the cells have produced all of the protein that they're going to, so we've separated them from the solution containing the protein. Now we're going to take that solution and purify it down to the protein that's specific to our medicine. This equipment is used to perform liquid chromatography, just one of the many steps that we use to purify the solutions containing our medicines. After that, we collect our protein again to perform our next unit operation. At Amgen, our workforce is committed to the highest quality standards because we know our products are going directly to patients. We collect samples to submit to our labs for testing at every step of the process.
This is the sample received from purification. Now we can take the sample into the lab for testing. We use advanced technologies to test our products to meet our own high-quality standards. Each sample has its own limits and specifications that have to be met before that sample can pass and the process can continue.
Hi, I'm Sharon. At this stage, we receive the bulk material. The bulk material gets formulated and placed inside tanks. The tanks are then transferred to the filling area where the product is filled inside vials or syringes.
Here at Amgen, we produce hundreds of thousands of product every day. And we ensure quality by utilizing automated and manual inspection. Once our product passes final inspection, it's ready for distribution around the world.
Fermentation is a reasonably simple process. A cell is selected based on its ability to produce the desired product. A seed stock of cells is put into a small amount of media. Media provides the nutritional products the cell needs to grow. When the population of cells has grown and consumed most of the nutrients, it's moved into a larger vessel with more growth media, and the process repeats. This scaling up is complete when the quantity of cells is large and healthy enough to transfer into a production vessel, often referred to as a bioreactor or fermenter. With plenty of fresh media now available and under tightly controlled conditions, the cells grow and manufacture product. When the fermentation is complete, the product is harvested. Fermentation is known as an upstream biotechnology process. It occurs early in the production flow, before recovery, purification, formulation, filling, and packaging. To better understand the fermentation process, we should first find out a little bit about the cells we use and what they may require to reproduce and stay healthy. Different cells have different needs. Some are aerobic, they need oxygen, while others are anaerobic and do not require oxygen. All cells require nutrition. A properly formulated media contains the necessary nutrients to allow cells to grow and produce. The fermenter mixes the cells evenly throughout the media to suspend the cells and supply the oxygen necessary for growth. Effective and efficient fermentation requires rigorous monitoring and control of the environment within the bioreactor. Key factors include temperature, pressure, pH, which is a measure of how acidic or alkaline the media is, oxygen, usually measured as dissolved oxygen within the media, and nutrient levels. Although the environment and the media are tailored to the needs of specific cells, the life cycle of almost all batches follows a predictable pattern. The growth pattern has 4 phases: lag, exponential or log, stationary, and death. When a cell is first introduced to fresh media, it has to adapt to its new environment. This creates a lull or lag in the growth timeline. After the organism adapts, the batch takes off. The cells begin dividing at a constant rate, an exponential or logarithmic or log increase, doubling, then doubling again, and on and on. As the nutrients in the media are consumed, toxic metabolic waste products build up, cells begin to die, and growth slows. When it reaches a point that just as many cells are dying as are dividing, the batch enters the stationary phase. This is the point at which the key nutrients are completely consumed, the fermentation is stopped, and the fermented broth is harvested. If the fermentation were allowed to continue, the cells would enter the death phase. More cells die than divide, and, similar to the exponential phase, the death rate increases logarithmically. Now that we have a basic understanding of how fermentation works, let's look at an actual process and see how it all comes together. For our sample process, we will look at the production of green fluorescent protein, or GFP. GFP is broadly used as a biological marker. It's a fluorescent dye that's very well tolerated by most cells and doesn't interfere with normal cellular function. In the GFP fermentation process, we'll need to add an antibiotic to protect the purity of the batch, and then, late in the process, a biochemical inducer to turn on the GFP gene. Our materials for this process will include a bacteria seed stock, in this case E. coli, that has been genetically enhanced to produce GFP. The basic ingredients for a compatible media, which include nutrients, stabilizers, an antibiotic, and an anti-foaming agent, and IPTG, which is the biochemical inducer that switches on the GFP gene. The equipment that we'll be using includes a 300-liter bioreactor, a UV-vis spectrophotometer to monitor the optical density, which is a measure of the concentration of cells in the bioreactor, a glucose analyzer to measure glucose, a key nutrient, an offline pH meter to help track the acid-base balance and adjust online measurements if needed, and a broth tank for our final product. The bioreactor is equipped with a water jacket around the vessel to regulate temperature, and integrated sensors to monitor key environmental factors, including dissolved oxygen, pH, internal temperature, water jacket temperature, and vessel pressure. The reactor also has an agitator, dedicated ports for adding seed stock and media ingredients, separate ports for acid and base supplement, air filters for supply and exhaust, and valves for drawing samples and for harvesting. Most fermentation and monitoring functions can be managed from the bioreactor's dedicated process controller. Before the fermentation process can begin, the area must be prepared. Preparation includes removing equipment and material that won't be used in the process, cleaning and sanitizing the area and equipment, and sterilizing equipment as required by the SOPs, Standard Operating Procedures. Sterilization is used to eliminate unwanted microorganisms which can grow naturally in the fermentation media and process equipment. Also, all required materials and documentation should be gathered and prepared, and all process control software should be loaded and verified. The fermentation batch process will be guided and documented with the BPR, Batch Process Record. The batch record leads the operator through the process, step by step, with each step requiring a sign-off and separate verification. This record also includes spaces for documenting key times, activities, and instrument readings. The GFP fermentation process really begins with the expansion of our bacteria seed stock. After removing the specially modified E. coli from the freezer and thawing it, it's used to inoculate a small amount of fresh media in a shaker flask. After the number of cells has reached the target amount, the thriving cells are ready for fermentation. Meanwhile, in the fermentation area, operators begin with a complete check of all critical equipment. Valves, caps, and lines are checked. Hoses are tightened. Probes are verified and calibrated. And 10 kilograms of HPW, high purity water, is added to the vessel. The bioreactor is brought up to normal process pressure and held there in order to check for leaks. The pressure is monitored over a 30-minute period. If a leak is detected, the problem is corrected and the test is run again. Once the reactor passes the test, we are ready to mix the media in the vessel. The agitator is turned on, and the ingredients are added. Yeast extract, tryptic soy broth, ammonium chloride, sodium biphosphate, monopotassium phosphate, and an anti-foam compound. Once all the initial ingredients are in, another 10 kilograms of high-purity water is added. All ports and valves are closed. All condensate valves are opened. And the bioreactor begins an SIP, sterilize in place cycle. The target for sterilization is 121 degrees Celsius for 30 minutes. As soon as the temperature climbs to the targeted temperature, the condensate valves are closed, and the SIP cycle completes automatically. Both the vessel and the media are now sterile, and we're ready to add the final ingredients to our media. The glucose hose is attached to the vessel, the connection is steamed to sterilize it, and the separately sterilized glucose antibiotic solution is pumped into the vessel. Then a manual pH reading of the media is taken, and the bioreactor is set up for its fermentation cycle. After the inoculation hose is connected to the reactor and steamed for 20 minutes, the expanded seed stock is pumped into the reactor containing the media. Fermentation now begins. The operator takes 0-hour readings and begins to regularly monitor batch temperature, agitator RPMs, dissolved oxygen levels, pH, vessel pressure, optical density, airflow rate, and glucose concentrations. Optical densities and glucose concentrations are of particular interest, so they're graphed as well as documented. When the targeted levels of glucose and optical density are achieved, it's time to add IPTG to the vessel to activate or turn on the expression of the green fluorescent protein in the cells. After allowing enough time for the cells to produce green fluorescent protein, usually 5 hours more, final readings are taken, and a sample is drawn to check the percentage of cell solids. The product is now referred to as broth. The broth, which contains spent media and cells, is complete when the key nutrient, glucose, is mostly consumed and the batch has reached the desired concentration. The batch is then cooled down, pumped into a broth tank, and labeled with the batch number, volume, time, and date. The fermentation process is now complete. The harvested broth will now move downstream to the recovery process, where the cells will be ruptured to free the green fluorescent protein, and the protein will be separated from the other broth components. Biotechnology companies rely on fermentation to produce their products. In the typical process, a particular microorganism is used to produce a valuable compound or product. Fermentation permits us to scale up that production. Instead of a single cell producing our molecule of interest, we use hundreds, thousands, and then millions and millions of these tiny microfactories to produce many different products on a commercial scale. In fermentation, there are two ways in which the product of interest is produced by the cells. Extracellular production is when the cell secretes the product of interest. Examples of this could be certain amino acids, enzymes, or monoclonal antibodies. In the case of extracellular production, the biomass or cells are discarded at the beginning of separation, and the medium is kept for further processing. Intracellular production is when the product of interest is produced by the cells and kept inside the cell. And during the early stages of separation, the biomass is collected and then disrupted so that the product is released. In our treatment of fermentation, we concentrate on intracellular fermentation. But fermentation is only the beginning of this journey. Let's look downstream at the next process step in the production flow, recovery. The final product of fermentation is called broth. Broth contains the molecule of interest. However, the molecule is still locked inside a host cell, and millions of these host cells are suspended in a pool of depleted media and metabolic waste products. In the next step, recovery, we perform two important functions: separating the cells, solids, from the broth liquid, and separating the molecules of interest from their host cells. Exactly where within the production flow those technologies are employed varies significantly from product to product. Recovery processes commonly use a variety of tools such as centrifuges, cell disruptors, and microfiltration to isolate the product. In this program, we are going to look at a typical recovery process used in the manufacture of GFP, green fluorescent protein. GFP is broadly used as a biological marker, which, if attached to a drug, could provide researchers with a visual story of where the drug goes. It's a fluorescent dye that's very well tolerated by most cells and doesn't interfere with normal cellular function. So how does recovery work, and how does it fit into the total production flow? Here's a very basic overview. After fermentation is complete, the broth is harvested and sent to recovery. Here, the E. coli host cells are separated from the liquid broth, then suspended in a new solution to wash the cells, separated again, resuspended in a new solution, homogenized to break open the cells. The cell debris is removed. Once recovery is complete, the product is sent to purification to be refined and concentrated. But to really understand recovery, we'll need to closely examine our process so we can appreciate what each step accomplishes and why it's important to the process. Let's start with the tools we'll use in the green fluorescent protein recovery process. Those would include a disc stack centrifuge to separate solids from liquids, a homogenizer to break open the E. coli cells, and a 0.22 micron filter to separate any remaining solids in the product solution. Our materials include the broth from our fermentation process, high-purity water that has been reverse osmosis filtered, deionized, and UV sterilized, and a buffering solution to help stabilize the pH of our product and keep it in suspension and prevent the product from degrading. The recovery process is managed through the use of a batch process record. The batch record leads the operator through the process, step by step, with each step requiring a sign-off and separate verification by a second operator. This record also includes spaces for documenting times, activities, operation steps, and instrument readings. Before the process can begin, the recovery area must be cleaned and organized. Any unnecessary equipment or materials should be removed, and the area must be cleaned and disinfected to decrease the level of microorganisms. All equipment must be cleaned, sanitized, and set up as required by standard operating procedures. All required materials and documentation must be gathered and prepared. And any updates to the process control software should be made and verified. GFP recovery begins with the arrival of the broth tank. A sterile hose is run from the broth tank to the disc stack centrifuge, and the tank is pressurized to drive the broth into the centrifuge. After the centrifuge has reached a stable running speed, the inlet valve is opened and broth enters the bowl. The centrifugal force of the rotation forces the denser material, the solids, to the sides of the bowl, much like the spin cycle on your clothes washer, while the liquid flows through and out of the centrifuge. As more broth enters the bowl, it displaces the now clarified, solids removed, liquid to the top of the bowl where it exits, while the cells continue to build up on the bowl surface. The centrifuge has an integrated RPM monitor. If the unit is not rotating at a stable running speed, the controller will alarm and shut it down. The liquid leaving the bowl is known as the clarified stream because almost all the solids have been removed. A sensor monitors the clarified stream for percent solids. When this value rises, it indicates that the bowl is at capacity and the solids must be removed before processing more broth. The solids are the E. coli cells, and they contain the product. When the bowl has reached capacity for solids, the bowl opens and the solids are discharged into an appropriate container for collection. Once the solids are discharged, the centrifugation step can resume, while the clarified liquid is waste. At this point, the cells are in a paste form, and although most of the liquid has been removed, our cell paste is still about 40% liquid by weight. The remaining liquid contains high levels of metabolites and salts that could complicate downstream processing, so we're going to lower those levels by washing the cells. The cell paste is suspended in a buffered solution and then run through the centrifuge again. As the clarified liquid leaves the centrifuge this time, it carries many of the contaminants from the fermentation step with it. The cells, once again in paste form, are ready for the next step, cell disruption, also called lysing. The cells are resuspended in a buffered solution and then pumped at high pressure, 900 bar, which is about 13,000 pounds per square inch, through the homogenizer. Inside the homogenizer, they are forced through a tiny orifice. Just like a balloon being tightly squeezed, the cells can't take the stress, and they rupture and break apart. And to ensure that all the E. coli cells are ruptured, the solution is cycled through the homogenizer a second time. After the second homogenization, the lysed cell solution is pumped back through the centrifuge. But this time, our goal is different. Before lysing, our product was held within the E. coli cells. Now, with the cells broken apart, the cell contents, including cytoplasm and green fluorescent protein, are mixed into the buffered solution. The centrifuge again spins out the solids, which are primarily cell debris, and it's the clarified liquid which contains our product, GFP. This time, we discard the solids and keep the liquid, which is now known as lysate. Although the centrifuge has removed almost all of the cell debris, some small particles still remain. We'll remove those with our final recovery process step, filtration. The centrifuge lysate is pumped through a 0.22 micron filter. This filter is fine enough that it removes virtually all of the remaining solid materials. At this point, the process stream is referred to as clarified lysate. The recovery process is finished. The clarified lysate is pumped into a vented, temperature-controlled transfer vessel. This lysate tank then moves downstream to the next series of process steps, purification, where dissolved impurities are removed from the GFP solution and GFP is subsequently concentrated and stabilized.